Hey there, deep divers. Have you ever considered that some of our most, established medical treatments might have surprising hidden powers we're only just beginning to truly understand?
It happens more often than you might think.
Well, today, we're taking a deep dive into just that, a fascinating revelation about conventional chemotherapy, often viewed as, you know, powerful but maybe a bit blunt.
A blunt instrument, yeah.
We're discovering it's actually a sophisticated conductor of our body's own immune system in the fight against cancer.
It's truly a paradigm shift. For decades, honestly, the focus was almost entirely on chemotherapy's direct ability to, kill cancer cells, full stop.
All right.
But now, cutting-edge research is uncovering its profound and often unexpected capacity to actually stimulate an anti-cancer immune response.
Exactly. So our mission today is to unpack this incredible story. We'll explore how these old drugs are becoming new again, sort of working hand in hand with our body's defenses.
And what that means for combining them with newer treatments like immune checkpoint inhibitors.
Precisely. We're drawing from some really groundbreaking research here that challenges our fundamental understanding of cancer treatment. So let's get started and see what this means for you.
Okay, let's unpack this. Historically, when we thought about chemotherapy, the traditional image is often of that blunt, non-specific weapon. What was the accepted view for so long?
Traditionally, chemotherapy agents were identified simply because they could inhibit cancer cell growth or cause cell death, usually tested in lab dishes, in vitro or in mice without working immune systems.
Immunodeficient mice, right?
Exactly. So these drugs were indeed pretty nonspecific. They targeted fundamental processes like DNA replication or repair things hyperactive in cancer cells.
It's like trying to stop a runaway train by just cutting the main power line. It affects a lot of other things, too.
That's a good analogy. And that nonspecificity explains why we often see side effects in other fast-growing tissues, like hair follicles, hence hair loss, or the gut lining causing those digestive issues.
So our healthy, rapidly dividing cells get caught in the crossfire. Precisely. Collateral damage. But the thinking was, cancer cells, with their crazy high growth rates, often faulty repair systems, and just inherent stress levels, were preferentially vulnerable. They were already sort of teetering on the edge, chemically speaking.
And chemo just pushed them over.
Right. But here's where it gets really interesting. For decades, this whole other piece of the puzzle, our immune system, was largely overlooked in this context.
A powerful ally just sitting on the sidelines, ignored.
Systematically ignored, you could say, yes, until maybe the mid-2000s. But then clinical observations started hinting that some of these agents actually stimulated, or at least were compatible with, efficient anti-cancer immune responses.
Hints in practice.
Yeah. In a subset of patients, these immune effects seem to provide therapeutic benefits beyond the initial treatment window. The positive effects lingered longer than you'd expect from just cell killing.
So an underlying immune effect was there all along, even if we didn't fully recognize it. How did researchers finally pin this down? What was the key evidence?
Well, there's now an abundance of preclinical studies, really over the past 2 decades. They showed that many conventional chemotherapies, think anthracyclines, oxaliplatin, taxanes, are far more effective in mouse models with intact immune systems compared to those lacking them.
So the immune system makes a big difference in how well the chemo works.
A huge difference. And notably, this difference is often most pronounced when chemotherapy is given at doses below the maximum tolerated dose, the MTD, or in these sort of metronomic schedules, lower doses given more frequently.
Interesting. So it's not just about hitting cancer cells as hard as possible.
Exactly. It suggests something more subtle is happening, a beneficial effect beyond just transient cell killing. It points towards, engaging the immune system.
Okay, quick recap then. We've shifted from seeing chemotherapy only as a direct cell killer to realizing it's also acting as a kind of subtle immune recruiter. We're looking at these familiar drugs with completely new eyes.
That's the core idea. The therapeutic efficacy of many conventional chemotherapies involves a significant immunological component that we just didn't appreciate before.
Okay, so if chemotherapy is an immune recruiter, how does it specifically make cancer cells more visible to our immune system? Like, how does it turn up the volume on that distress signal?
Right. For your immune system to launch a proper anti-tumor attack, 2 fundamental things need to happen at the same time. First, the cancer cells need to display antigens. Think of these as unique flags that your T cells can recognize.
Flags that say, I'm abnormal.
Essentially, yes. Second, they need to deliver adjuvant-like danger signals to specialized immune cells called antigen-presenting cells, or APCs. These signals are like a loud alarm screaming, danger, investigate this cell.
Got it. Antigens and danger signals. Let's break down the antigens first. How does chemo boost this antigenicity, make those flags more obvious?
Well, cancer cell antigens can arise from various things, maybe viral infections linked to the cancer, or new proteins created by genetic mutations, these neoantigens. Sometimes they re-express proteins normally only seen during fetal development.
Oncofetal antigens.
Exactly. Chemotherapy can potentially enhance this antigenicity in a few ways. It can force cancer cells to upregulate MHC class I molecules. Those are the platforms on the cell surface that actually display the antigens to T cells.
Like putting the flag on a taller pole.
Good way to put it. Chemo can also alter gene expression or cause more genetic defects, changing the whole spectrum of peptides or flags being presented. For instance, some drugs increase components needed to build those MHC platforms. It helps shine a spotlight on the identifying markers.
Okay, and the second part, the adjuvanticity, the danger signals. How do dying cancer cells actually shout for help effectively?
This is where something called damage-associated molecular patterns, or DAMPs, come into play. These are signals released by cells that are stressed or dying, even without an infection. They're basically internal alarm bells. Many conventional chemotherapies boost both antigenicity and, maybe more significantly, this adjuvanticity by inducing specific immunogenic forms of cellular stress and death. It's not just any cell death. It has to be a kind that rings the immune alarm bells.
And there's a special term for that kind of alarm-ringing death, isn't there? Immunogenic cell death, or ICD. What's the deal with ICD?
ICD is a specific regulated way for a cell to die that, crucially, enables your immune system to kickstart an adaptive response against whatever caused the death, in this case, the cancer cell antigens. It happens when stressed cells expose or release very specific DAMPs.
Okay, like what kind of damps?
For instance, a molecule called calreticulin, CALR. Normally it's inside the cell, but during ICD it moves to the cell surface. It acts like a bright eat me signal, telling immature dendritic cells, those key APCs, to engulf the dying cancer cell.
So it invites the cleanup crew that can also sound the alarm.
Precisely. Then there's ATP, the cell's energy currency. When it's released outside the cell in large amounts, it acts like a chemical flare, recruiting more DC precursors to the tumor site and triggering local inflammation.
Draws more immune cells in.
Exactly. Also, nucleic acids, like DNA and RNA, if they end up in the wrong place, like outside the cell or loose in the cytoplasm, they can trigger innate immune sensors. This leads to the production of type I interferons, a powerful antiviral signal. It's often called viral mimicry.
Viral mimicry. So the dying cancer cell sort of tricks the immune system into thinking there's a virus.
In a way, yes. It activates similar warning pathways. And other proteins like HMGB1 get released too, and they help the dendritic cells mature and become fully active antigen presenters.
So all these signals tell the dendritic cells, engulf this dying cell, process its flags, antigens, and go tell the T cells to hunt down anything else with these flags.
That's the essence of it. The DCs cross-present the tumor antigens to cytotoxic T lymphocytes, your killer T cells, which then patrol and destroy residual cancer cells. So it's profoundly important how the cancer cells die.
It really is. And you mentioned earlier that the mechanism causing cell death isn't always the same as the one causing ICD. That's a critical point. How do researchers figure out which chemo drugs are good ICD inducers?
Right, they aren't always linked. For example, doxorubicin and oxaliplatin kill cells by messing with DNA synthesis, but they also trigger separate stress pathways in the cell's cytoplasm that lead to ICD. And interestingly, drugs that look similar chemically don't always act the same immunologically. Oxaliplatin is a much stronger ICD inducer than the closely related cisplatin.
Huh. So you can't just guess based on structure.
Not reliably, so researchers use drug screening techniques. They treat cancer cells with different drugs and then measure the release of those key DAMPs like CALR on the surface, ATP release, HMGB1 release. They look for that specific shout profile.
And I read technology is helping here too. There's even an algorithm.
Yes, that's quite exciting. Machine learning algorithms have been developed. They analyze data from these DAMP screens and learn to predict the probability that a given drug, based on its chemical properties, will induce ICD. Wow. This ICD prediction algorithm has been tested and validated. It helps us classify drugs confirmed inducers like oxaliplatin and ampercyclins, potential candidates like cisplatin or paclitaxel, or likely non-inducers. It's a really valuable tool for designing smarter combination therapies down the line.
Okay, so we've covered how chemo can make cancer cells light up for the immune system through ICD. But you also mentioned it can act more broadly, right? These off-target effects on the immune system itself.
Absolutely. When chemotherapy is given at the right doses, often below or near the maximum tolerated dose, it's frequently not globally immunosuppressive. In fact, many agents can directly interact with various immune cells throughout your body, tweaking their function to help fight the cancer.
So it's not just hitting the tumor, it's also fine-tuning the immune orchestra systemically.
Exactly. It's modulating the immune environment.
Like how? What specific immune cells are we talking about?
Well, one really key mechanism is the selective depletion of immunosuppressive cells. Think of your immune system as having accelerators and brakes. Some cells act as brakes, preventing overactive or autoimmune responses, but in cancer, they can unfortunately protect the tumor.
Like regulatory T cells, Treg cells.
Treg cells are a prime example, yes. Also, myeloid-derived suppressor cells, MDSCs, and certain types of tumor-associated macrophages, the M2-like ones. Some chemotherapies can preferentially kill off these suppressor cells. Low-dose cyclophosphamide is famous for depleting Treg cells, which can basically take the foot off the immune brake, allowing a stronger anti-tumor response.
Removing the brakes.
Yeah.
Clever. What about hitting the accelerator? Activating the good guys?
Exactly. Chemotherapies can also activate immune effector cells. We already mentioned dendritic cells, DCs, being crucial for priming T-cell responses, especially after ICD. Agents like cyclophosphamide and gemcitabine can stimulate DC recruitment and activation.
Building on those danger signals.
Right. And some drugs, like 5-fluorouracil or oxaliplatin, seem to directly nudge DCs towards activation, boosting their production of helpful signals like IL-12 and reducing suppressive molecules like PD-L1 on their surface. Pemetrexed, another drug, has been shown to enhance the activation and function of T cells themselves.
So it's a multi-pronged approach, removing suppressors, activating effectors. Now, if we zoom out even further, how does chemo influence the entire body's physiology? This sounds like it goes beyond just immune cells.
It absolutely does. We're seeing profound whole-body immunomodulatory effects. It influences the whole dialogue between you, the host, and the cancer. Two really fascinating areas are the gut microbiota and neuroendocrine pathways.
The gut microbiome. How does chemo affect our gut bacteria in relation to immunity?
Well, most chemotherapies, unfortunately, have some gastrointestinal toxicity. This often leads to dysbiosis, significant shifts in the composition and balance of your gut bacteria. And we now know your gut microbiota plays a fundamental role in setting your baseline immune tone.
So messing with the gut bacteria can mess with immunity.
It seems so. Dysbiosis has been linked to reduced immunological competence, and there's evidence that it can reduce the efficacy of some chemotherapies and even ICIs. For example, studies have shown that broad-spectrum antibiotics, which wipe out gut bacteria, can blunt the effectiveness of certain chemo regimens.
Wow. That implies maybe managing the gut microbiota could actually help treatment.
That's the thinking. Modulating the gut microbiota with things like prebiotics or probiotics is an active area of research, potentially to improve treatment efficacy and maybe even lessen some toxicities. It's a really hot topic.
Okay, and the other area? Neuroendocrine circuitries. That sounds like stress hormones.
Exactly. The psychological and physiological stress associated with the cancer diagnosis and the treatment itself can elevate your body's levels of endogenous glucocorticoids, basically, stress hormones like cortisol.
And that's bad for immunity.
Generally, yes. High cortisol levels can cause systemic immune suppression. There's even evidence this stress response can directly abolish the anti-tumor efficacy of certain chemotherapies like anthracyclines or oxaliplatin by changing the immune cells within the tumor microenvironment.
So stress can literally make the treatment less effective.
It appears so, and it raises questions about the common practice of giving patients exogenous glucocorticoids like dexamethasone to manage side effects. Could they also be dampening the chemo's immune benefits? It's complex. Interestingly, some retrospective studies have hinted that using nonspecific beta blockers like propranolol might actually improve patient outcomes, possibly by counteracting some of these stress-related immunosuppressive signals.
That's fascinating. Our mental state, our stress levels, our gut bacteria. It's all connected to how chemo works immunologically. Okay, let's pause for another quick check-in. We've journeyed through how chemo makes dying cancer cells shout for help via ICD, how it can fine-tune specific immune cells by removing brakes and hitting accelerators, and even how it interacts with our gut microbes and stress responses.
Right. Understanding these diverse mechanisms, both the direct on-target effects on cancer cells and these broader off-target or systemic effects, is absolutely crucial as we think about designing smarter, more effective cancer treatments, especially combinations. #4. chemoimmunotherapy in the ICI era, and the road ahead.
Which brings us neatly to the next point. Given everything we've just discussed, it seems almost obvious to combine chemotherapy with immune checkpoint inhibitors or ICIs. What's the strategy there? It feels like bringing two powerful approaches together.
It is. ICIs, which target pathways like PD-1, PD-L1, or CTLA-4, have been revolutionary on their own. They achieve really impressive response rates, maybe 20–40%, in many solid tumors where we previously had few options.
But they don't work for everyone, and they have their own side effects.
Exactly. Immune-related adverse events can be significant. So the goal of combining chemotherapy with ICIs is ideally to get the best of both worlds. Early disease control from the chemotherapy's direct killing effect, plus durable long-term control from the ICI unleashing the immune system. You aim for synergy, the combination being better than either agent alone with manageable side effects.
And this is already happening.
Oh, yes. Chemoimmunotherapy has become the standard of care in several cancer types, like certain lung cancers and bladder cancers. And there are, I think the number is over 450 clinical trials currently ongoing, exploring or optimizing these combinations across many different cancers.
It makes sense. It's like chemotherapy is heating up immunologically cold
Yeah.
tumors that don't have many immune cells in them, making them ready for the ICIs to then really do their job.
That's a perfect way to describe the rationale. ICD-inducing chemotherapies can, in theory, convert these cold, non-inflamed tumors into hot lesions, packed with dendritic cells and those killer T cells. This primes the pump, making the tumor much more likely to respond to an ICI which needs those T cells present and active.
And we see evidence for this?
We do, consistently in preclinical mouse models. And clinically, we also see that chemotherapy can sometimes induce the expression of PD-L1, the very target of many ICIs, on cancer cells or surrounding immune cells. This upregulation, often driven by those type I interferons we mentioned earlier, is itself a biomarker suggesting the tumor might now be susceptible to ICI therapy.
Can you give us some concrete examples from trials or guidelines? Where is this combo making a real difference for patients?
Sure, a great example is in non-small cell lung cancer, NSCLC. Current guidelines often recommend chemoimmunotherapy, usually a platinum doublet like carboplatin plus pemetrexed, combined with an anti-PD-1 or anti-PD-L1 ICI right up front for many patients, particularly those whose tumors have low levels of PD-L1 expression.
So chemo helps, even when the main ICI biomarker is low.
Exactly. And trials like the Pacific study showed a huge overall survival benefit for adding durvalumab, an anti-PD-L1 antibody, after patients had completed chemoradiotherapy for stage 3 NSCLC. Another one, the Checkmate 9LA trial, showed that giving just a couple of cycles of chemo along with 2 ICIs up front improves survival in advanced NSCLC compared to chemo alone, importantly, regardless of the patient's PD-L1 status.
So strong evidence supporting the idea that chemo can make tumors more responsive to ICIs.
Yes, findings like these strongly support the notion that certain chemotherapies agents like doxorubicin, 5-FU, oxaliplatin, platinum drugs can indeed sensitize tumors to immune checkpoint blockade.
Okay, so combination therapy is clearly a major path forward. But it sounds like despite the successes, there are still quite a few unknowns, right? We're still figuring out the best way to conduct this complex immune orchestra.
Absolutely, without a doubt. While the clinical effort is massive and ongoing, several critical open questions remain.
Such as,
Well, first, the nuance of ICD. We need a much more precise understanding of how different DAMPs and cellular stress responses really contribute. Sometimes, under certain conditions, a DAMP might actually be immunosuppressive, or its effect could change depending on how or when it's released. It's not always black and white.
okay, the details matter.
They really do. Second, other immune checkpoints. Most focus has been on PD-1 and CTLA-4, but there are many others, LAG-3, TIM-3, VISTA, NKG2A. We have very limited information on how chemotherapy affects these pathways. Targeting them alongside chemo might reveal novel synergistic regimens.
We need better biomarkers too, I imagine.
Definitely. Better biomarkers are crucial. PD-L1 is useful, but it's often just a snapshot taken at diagnosis. It might not reflect the tumor microenvironment after it's been reshaped by chemotherapy.
Oh.
We need dynamic predictive biomarkers that tell us who will benefit most from which combination, not just prognostic ones.
And finding the best combinations and schedules.
Yes. Optimal combinations and scheduling. Should we combine chemo with agents that boost DC activation like FLT3 ligands or TLR agonists? What about DC vaccines? And the timing, should chemo come before, during, or after ICIs? What about dosing low-dose metronomic versus MTD? Preclinical data often favors low-dose schedules for immune stimulation, but translating that reliably to the clinic is still ongoing and seems to vary by cancer type and drug. Lots of trials are tackling this.
And finally, how do we know if it's true synergy?
That's the million-dollar question. True synergy versus patient heterogeneity. Is the benefit we see in trials because the drugs truly work better together? Or is it just that the combination covers more bases, hitting patients who would have responded to chemo or ICI individually? Distinguishing that requires really detailed analysis of patient samples looking at mutations, gene expression, immune cells over time, coupled with very smart clinical trial designs. Hashtag tag tag outro.
Wow. Okay. What an incredible journey through the unexpected, intricate world of chemotherapy's immune effects. It's so clear that this isn't just a blunt weapon anymore. It's emerging as a really sophisticated immune modulator, capable of orchestrating an anti-cancer response from within.
Indeed. And this realization has two huge implications for you, for everyone listening. First, it highlights that a patient's own immunological health and competence profoundly influences how well chemotherapy is likely to work.
Your immune system matters for chemo outcome.
Absolutely. And second, it provides incredibly solid scientific grounds for developing these novel, synergistic therapeutic regimens that combine conventional chemotherapy with modern immunotherapy like ICIs.
It's just a powerful reminder, isn't it, that our understanding of biology is constantly evolving. And sometimes the biggest breakthroughs come from looking differently at treatments we thought we already fully understood, finding secrets hidden in plain sight. It makes you wonder, as we keep uncovering these intricate connections, what other old standard treatments might hold similar hidden immunological secrets, just waiting for us to unlock them?
That's the exciting frontier. This ongoing research really promises to help us maximize the benefits of immunogenic chemotherapy in this era of immune checkpoint inhibitors, hopefully benefiting a much larger fraction of patients fighting cancer.
Absolutely. So as you reflect on this, maybe just consider how deeply intertwined all our body systems truly are. Our immune cells, our gut bacteria, even our stress responses. And how personalized medicine in the future will increasingly need to leverage your unique biology in the fight against cancer. Thank you so much for diving deep with us today.